I have, let's see, I'm talking to you guys, I believe, each day this week, except Friday. Um, And then we'll,
Be awesome. Finish that sentence, it will be
finish up
awesome.
discussion of the pathophysiology later on this week as well. So, spinal cord. These are the learning objectives, 4 simple little objectives, that's all. If you look at the anatomy of the cord, and a lot of this is going to be anatomy. Near the end, we'll talk about some neurocircuitry, but we're going to start here with anatomy. Remember that the spinal cord ends at about L2 vertebral level in the adult. If you look at the cord itself, it's gray matter surrounded by white matter. White matter is so called because it's mostly containing myelinated axons traveling up and down the cord, getting up and back to the brainstem and the higher cortical functions. Within the different segmental levels, you have the dorsal and ventral roots. Dorsal roots, or rootlets, enter into the dorsal horn of the cord. The ventral rootlets exit the ventral horn of the cord. There are different fissures and grooves in the cord. The most prominent one is the anterior median fissure, which is going to carry the anterior spinal artery. On the back side of the posterior aspect of the cord, there are 2 intermediate sulci. The posterior spinal arteries run in that in those intermediate sulci. There's a septum that divides the cord in the back, that's very prominent, divides it to a right and left half. And at the 6th thoracic level, at the 6th thoracic spinal cord level, not the vertebral level, at the 6th thoracic spinal cord level, there is a septum that you'll see right there that divides this posterior part into 2 sections. Below T6, you don't see that septum. It's only at T6 and above do you have that division. I'll explain how that is created here in a little bit. If we remind ourselves of the pia surrounding the cord, we've got the the meninges surrounding the cord. We have the pia that is adherent to the cord and to the rootlets themselves. They form the outermost layer of the nervous system. Next, you'll have the arachnoid, and beyond that will be the dura. On that posterior aspect up here, you see that little septum that I mentioned just a minute ago? Posterior intermediate septum. That divides that posterior part into 2 sections. Now the posterior part, the whole posterior part is called the posterior funiculus versus a lateral funiculus and an anterior funiculus. And if we look at this slide right here, you can see it more in a more stylized manner. Anterior funiculus, lateral, and posterior funiculus. Posterior funiculus is divided by that septum into 2 different parts. Yes, ma'am.
So each half has 2 little parts.
Yeah, so if you look across there, 1, 2, 3, 4. There are 4. Let me give you why I'm stressing this so much. Is that sensation from T6 and below is carried in this half. Sensation from T6 and above is carried in that half. So they have different names that you'll see in just a minute. All right, the gray matter is divided, Roman numeral, into 10 different regions. These are called Rexed, R-E-X-E-D, Rexed's lamina. 1 through 5 are the dorsal horn. 7 is this large area right here. 8 is around this area. 9 contains the motor cells that go to skeletal muscle. And 10 is around the central canal. Autonomics you'll find in 10. Now, the reason these have different numbers is because the synapses and the cellular architecture is different with each of these lamina. I don't care that you know the different Rexed lamina, other than there are 10. 9 contains motor cells, 10 is around the central canal, serves as autonomic function. Most of it dedicated to the receipt of sensory information. Another thing anatomically I want you to remember though is remember the pia is right here, it goes into the different fissures. Remember the denticulate ligaments that are laterally made of pia that connect to the underside of the arachnoid to help hold the spinal cord in place from lateral movement, denticulate ligaments. If you look at the vascular supply of the cord, the anterior, so to review real quick, you have the 2 vertebral arteries that go to form the basilar artery. Off of the vertebral arteries, they extend a branch medially. That's the anterior spinal artery, runs in the anterior median fissure. And in each vertebral artery will give rise to a posterior spinal artery, again, that runs in the intermediate sulcus down the backside of the cord. All right? There's the anterior spinal right there. Posterior spinal is back there. You have 2 posterior spinals, 1 anterior spinal. Off of those, you see quite an extensive arterial blood supply to the cord. Now, that's pretty much the way it goes, except when you get down here to the low thoracic region, the main blood supply, which is the anterior spinal artery, is pretty much used up. So we have to recharge the arterial supply, and that is, if you recall the aorta here, and you have the intercostal arteries that come off of the aorta, they send branches called the radicular branches into the spinal, to the vertebral canal, into the cord. There is on the left side, somewhere between T9 and L1, the intercostal or segmental artery that comes off the cord has a very large branch that dedicates itself to the resupply of the anterior spinal artery. So it dumps in there, like you see, goes up and it comes back down to reform or recharge or resupply with blood the anterior spinal artery. It's called the artery of Adamkiewicz or Adamkiewicz, or however you say that in Polish or whatever language that is. Very specific. It's only on the left side and it's somewhere in here. It varies between people. The reason this is important is you've got to, if you're doing the contrast studies and injecting I think I probably mentioned this in anatomy. You inject thick contrast material doing a renal arteriogram, you have to worry about dumping all of that thick contrast material right here and blocking that artery. Clogging that artery with thick contrast, because if you gork that artery, then that is essentially supplying the lower part of the spinal cord that supplies your legs. And you can wind up with paraplegia from messing with that artery. You can see it here quite well on these arteriogram. It goes up and down. You can see it on both of them. All right? So that's the importance about that artery. Now, parcellation of the gray matter. It says here, what I mentioned earlier, it's done so because not only do they look different, they have different functions. I mentioned these being large motor cells going to skeletal muscle, that's part of lamina 9. There's also two parts called the intermediolateral and the intermediomedial cell column. You know the intermediolateral cell column as the site of cell bodies for preganglionic sympathetics and parasympathetics if we get down there in S2, 3, 4. Right? That's where you find the lateral horn for the IML. The intermediomedial is there around lamina 10. That's where visceral afferent information, visceral sensory information goes back into the cord. That's where the relays occur for visceral sensory information. So visceral motor is out here, visceral sensory is in there. If you look at these motor neurons, they are certainly large multipolar neurons. You've got several dendrites in this picture here. There's one axon, they only have one axon. And here's that clearing area, that's the axon hillock. And I mentioned the other day that this granular substance was called Nissl substance. But I got to thinking, I don't think I mentioned what it was. It's rough endoplasmic reticulum. The motor neurons, or neurons, have a very extensive RER to make all the proteins and neurotransmitters and stuff like that. So that's what Nissl substance is rough endoplasmic reticulum. Now, a lot of what's known about neuroanatomy, it's amazing because it's been known for over 100 years, long before imaging studies were able to decipher what's going on precisely with the nervous system. But they've known that if I touch my right here, then they've known that where that reaches my cortex is right there. They've known that for 100 years. How? Well, in all of the people in all of the world with all of the different lesions from gunshot, stab wounds, accidents, after they died, they dissected their nervous system and did different types of stains to look at what was gone versus normal. So this is a type of staining. It's a myelinated fiber staining. A person who's had a stroke up there has knocked out the descending motor input to their spinal cord. So if you look, that's where the descending motor control of spinal cord is right there. It's gone, it's there on that side, gone on that side. Just comparative studies in people with lesions have done a great deal to mapping the circuitry of the nervous system. Right? Now, if you look at, this by the way is in the medulla, we're going to cover this tomorrow, so it doesn't look like the spinal cord. This though is the spinal cord, and if we look at it, and you dissect using those types of lesion experiments, the different areas of the spinal cord, you come up with a whole host of tracks. So the fibers that are arranged in the spinal cord are not just randomly arranged, they're in bundles. They travel in bundles. Another thing you need to keep in mind is that when I'm talking about the gray matter, understand that when you look at a section of gray matter, and I say this is lamina 10, contains motor neurons, understand that there's a whole column of lamina 10 that runs the entire length of the spinal cord. These things are in columns. They're not little sections. They're columns of neurons. That's what you need to, they're three-dimensional columns of neurons. Like they're three-dimensional columns or tracks of axons. There are 9,000 tracks in the white matter. Not 9,000. These are the predominant ones right here. There are more than these, though. These, though, are the, what down here is highlighted the ones that are most significant. And the ones that we're going to be talking about over the next few days. On the ascending pathway, so this is sensory of some type. Sensory of some type. You have the dorsal columns. The dorsal columns occupy the posterior funiculus. You see where it says gracilis and cuneatus, those are the two sections up here. The section that's closest to the septum, or the midline, is fasciculus. A fascicle is a group of axons. The fasciculus gracilis. The fascicle that's lateral is the fasciculus cuneatus. So when I talk about the dorsal column pathway carrying touch, if I touch you on the arm, you know that, as I mentioned earlier, this is lower extremity, that's upper extremity, that information is going to travel in the fasciculus cuneatus. If I touch you on the lower extremity, that information is carried over axons in the fasciculus gracilis. The way to remember that is, if that's the gonorrhea, GC. Put the G next to the septum, GC. You won't ever forget it. Which one's which? Okay? The lateral spinothalamic tract is out here in the lateral funiculus. The lateral spinothalamic tract is an ascending tract that carries pain and temperature. We'll go into the exact circuitry later on. Spinocerebellar tracts, and by the way, a lot of neuro, if you stop and pay attention to it, it tells you where it came from and where it's going. Spinothalamic starts in the spinal cord, goes to the thalamus. Starts in the spinal cord, goes to the cerebellum. The cerebellum, as we'll talk about on Thursday, does a lot towards the timing of movement. So a lot of proprioceptive information is traveled over the spinocerebellar tracts. On the descending side, the predominant fiber tract for volitional movement, I want to pick up that telephone. Your ability to execute that movement is carried over the cortico-spinal tract. Cortico, cortex to the spinal cord. There are two corticospinal tracts. The largest one is the lateral corticospinal tract, and we're just going to stick with that one. The ventral corticospinal tract is a very small tract that we're not even going to pay attention to, okay? But you're going to see it in a lot of slides. I don't want you to... Lateral corticospinal tract, that's the one that's important. Now, rubrospinal tract, rubro meaning red, comes from a red nucleus and goes to the spinal cord. It facilitates flexion, particularly of the upper extremities. So, if I'm going to pick up that telephone, I've got to have my corticospinal tract to do the volitional movement, but the rubrospinal tract helps refine that movement of the upper extremity flexors. Upper extremity flexors. The vestibulospinal tract, where do you think that comes from? Vestibular nucleus. The vestibular cochlear nerve, right? Not the cochlear part, just the balance part. Going to the spinal cord. What do you think it's involved with? It's involved with, get this, anti-gravity muscles. It keeps you upright. It helps keep you upright. What keeps you upright? Not the flexors, but the extensors. So the rubrospinal tract facilitates flexors, the vestibulospinal tract facilitates extensors. See, neuro is so easy. All right? Now, if you look at the spinal cord at different levels, it looks differently. You have two particular enlargements. One is a cervical enlargement, and one is the lumbar enlargement, because you've got an enormous number of neurons that have to go to supply the upper and lower extremities. That's what makes the enlargements. If you go through these different, these are all at the same magnification, you can see that there is quite a bit of difference between the gray matter and the white matter. For example, once you get up in the cervical region, right here, you're going to have a huge amount of white matter. Why would you have a huge amount of white matter? Because up here in the cervical region, all of the fibers from all of the dermatomes are going up and then coming back down. The most information going in and out of the brain is going to be found at the cervical level. Also, if you're at the low cervical level, which is where this one is, where does that say it is, about C8 or something like that? You're going to have a huge ventral horn because what's the brachial plexus? C5 to T1. That's where you're going to have a large horn. Are you going to have a lateral horn? No, because the IML, the autonomics, the sympathetics, where do you find the sympathetic preganglionics? Between T1 and L2. So in the cervical region, you're not going to have a lateral horn. You will have a prominent cuneatus and fasciculus here. This is above T6. I said it was above T6, right? I get below T6, there is no fasciculus, cuneatus, or gracilis. It's just the dorsal funiculus. This would actually, if you had to be precise, it would be the fasciculus gracilis, because that's the lower part of the body. But at this point, you just essentially call it the dorsal column. You also have, in the thoracic region, very, very, very tiny dorsal horns and tiny ventral horns. Why? Because the only thing that they're supplying or receiving information from is an intercostal space. Because we're between T1 and L2, you do have a lateral horn. There's the lateral horn, or IML. You go lower down, now you've got all of these motor neurons going to the lower extremity, which is bigger in most people than the upper extremity. That would look odd if your lower extremity was smaller than your upper extremity. Think about that for a minute. Anyway, you have a large ventral horn. You also have a lot of, white matter, but in the thoracic and the lumbar region, because you're not at the bottom of the cord yet. Once you get down into the sacrum, wow, look at this gray matter down here. Look at the small amount of white matter. Why do you have a small amount of white matter? Because all you're doing is, from above, you're supplying these dermatomes, and you're only sending the information up from these dermatomes. Not a lot left. So in terms of the white matter, there's not a lot left. The gray matter looks disproportionately large. Why? Because you've got all of these lower abdomen and pelvic organs you're supplying. In addition to the sensory components, and the pudendal nerve is S234, big time. Gray matter. And if you look at the motor neurons, if you look at lamina 9, there are two types of motor neurons that you're going to encounter. The predominant type will be somatic efferent fibers. Somatic efferent. These are going to muscles. Now, there's another fiber type that you'll find in lamina 9. These are called gamma efferent fibers. These are going to muscle spindles. You remember from physiology what a muscle spindle is. Embedded within the muscle itself, there are these little things called muscle spindles that send information back to the cord on how contracted the muscle is. They're supplied by the gamma fibers, and they're called intrafusal fibers, the two muscle types that are inside a muscle spindle. The gamma neurons, the motor neurons going to the muscle itself are called extrafusal muscles, and they're given the designated alpha motor neurons. Alpha motor neurons. Those are the predominant cell type. You would have, say 100,000 of these to one of these, if you were to look at ratios. Alpha motor neurons are the predominant type. Now, muscles have to be directed to contract. They cannot contract on their own. And the thing that you're all aware of is the simple reflex arc. So sensation comes in from somewhere that tells you that you need to activate that muscle. If I were to pick up Meredith's cell phone and it's actually 400 degrees, and oh, that's hot. So sensory information comes in from the skin, for example. It comes in from the muscle spindles, because I have to know how contracted my muscle is. That information goes in through the dorsal horn. Remember, all sensory information goes in through the dorsal horn. It's connected usually through interneurons with these gamma and alpha motor neurons that allow me to quickly withdraw my hand. Now, if I'm going to withdraw my hand, I'm flexing. So these have to be facilitated. What has to be inhibited would be the extensors. Simple reflex arc. Any questions about that? All right. We'll move it along.
What was the difference, sorry, between the extrafusal and intrafusal muscle fibers?
Extrafusal muscle fibers would be your biceps. The intrafusal fibers are the bag fibers and the nuclear chain fibers that are inside the muscle spindle itself. This is the muscle spindle, and that thing is embedded in the muscle itself. So that lets you, there's proprioception. It lets you know, along with receptors and tendons, how contracted things are. If you contract the muscle, you have to also contract this to maintain tension on it. If you just contract the muscle, and not this, then it becomes not taut. So it's not able to give you any information. So you have to maintain tension on the intrafusal fibers, just like you do the muscle. Okay? If you look at dermatomes, and this is just a refresher here, if you look at the upper extremity, you're arranged like a worm from top to bottom in a dermatomal, or in this case, a myotome pattern. So these are, and you don't certainly need to memorize any of these because you already know them from anatomy. So all the way down from a myotome arrangement. So it's essentially C4, 5, 6, 7, 8, T1. That's it. And whatever muscles are underneath there. C4, what do you think C4 gets? Deltoid. C5, what gets C5? Oh, biceps. Look at the chart. Am I right? Of course I'm right. It's that simple. Okay? It's a little bit more difficult with the lower extremity because your lower extremity started out like that. You had to rotate your lower extremity, so it kind of twisted the myotomes and dermatomes. So what was in the front in your lower extremity actually started out embryologically in the back. So you got reversed there. So just stick with, that's why I just have the upper extremity. I don't know the lower extremity. Okay? All right. Motor column comparisons. This is interesting because what do the different layers of the spinal cord have in common? They all have in common a motor, in terms of motor supply. They all have in common the motor supply to the axial skeleton. They all have in common the motor supply to your erector spinae, your semispine, your splenius, and all that stuff. So, where is the commonality in these cross sections? The commonality is in the medial part. When you get to the upper extremity and lower extremity, what we're going to do, we're going to put those motor neurons out here laterally. So if, understand? Follow that? All right, so if we talk about a motor neuron, as I said, muscles just don't contract. They have to be driven to contract. And this represents here an alpha motor neuron. Lamina 9, Rexid lamina 9 neuron. There's the axon going out to the muscle itself. When we talked about reflexes, here's facilitation for the flexors. There's the inhibition from the extensors, for example. But look how many descending tracts. So here's the corticospinal tract coming. There's your volitional movement right there. But look how many other tracts are involved in that neuron. There are a whole bunch of other tracts, some facilitatory, some inhibitory, to modulate that neuron. For example, when I reach, let's say I'm walking down these stairs. I'm walking down these stairs, and I've got all these motor neurons going to my lower extremity, right? And I'm not paying attention, and I step off of this step wrong. I misstep. What happens? I catch myself, right? I thought that the next step was going to be there, but suddenly it wasn't. My reflexes said, wait a minute, you're not where you're supposed to be, you have to do a correction. And the correction is, I don't fall down. But, you know, so when I don't fall down, I just do that, right? I weigh 225 pounds. If I expended all of my motor neurons to my leg, I bet I could push this disc that weighs more than 225 pounds. If I contracted everything in here, I bet I could do some damage to that disc. Why is it then, when I fall off of this step, why is it then I just catch 225 pounds and I not contract everything and do that? Because all of these things are modulating the degree of excitation that goes out instantly. And that's why. Everybody understand that little analogy? Because it's going to become important later on. Now, you call this neuron, the alpha motor neuron, you can say it's the final common pathway, because it is. All of this above input is meaningless without that, the alpha motor neuron. It's the final common pathway. If you lesion this, if you cut that axon, if you lesion the neuron, then that's called a lower motor neuron lesion. In order to do that, you have to lesion the ventral horn or the nerve itself. If you lesion all of this stuff up here, like if you have a transection of the spinal cord up here, you're cutting all that descending input below there, right? You call all of these things up here, not the spinal reflexes because they're local, but you call all of that up here the upper motor neuron. All right? So keep that in mind. If you lesion the alpha motor neuron or its axon, that's a lower motor neuron lesion. If you lesion anything above it, that's an upper motor neuron lesion. So if I had the spinal cord here, and I got a gunshot wound right there, I just produced two types of lesions. I produced a lower motor neuron lesion there and an upper motor neuron lesion, everything below it. Got it? Okay. Afferent fibers. Let's talk about the sensory tracts here for a minute. As we said, all sensory information comes in the dorsal root. And I've told you now about two systems. One is the dorsal column system. The other is the spinothalamic tract. This being touch, spinothalamic tract being pain and temperature. So here's how they enter the cord. The dorsal column pathway, which supplies, and this is written down somewhere else so you don't need to write it down. Kayla, if you shut your eyes, and if I say, point to exactly where I touched you just then. She pointed exactly to where I touched her. That is localized touch. Precise, fine touch. If I were to say, okay, Kayla, close your eyes. Close your eyes. And, no, actually, you're not a good one. You need somebody, Harry.
Right.
All right, close your eyes. All right, I want you to point to where I touch you. Look at there. He was able to point. I moved one hair on his gorilla forearm, and he was able to point right about where I touched him. That's very precise. One hair. That information is carried over the dorsal columns, as well as vibration, as well as proprioception. You don't need to write this down. It's written down somewhere else. And also the ability to, so if I take a dermatome and I say, Kayla, close your eyes. Am I touching you with 1 point or 2? So ability to distinguish 1 versus 2 point discrimination, that's carried over the dorsal column pathway. The cell bodies, remember from anatomy, of sensation below the mandible are located in the dorsal root ganglion. The peripheral process goes out, comes in, brings information in from the skin, joint receptors and stuff, enters the dorsal horn, and does not synapse, but ascends in the dorsal column. It ascends in the dorsal column. You call that neuron the primary afferent neuron. You can also call it a first order sensory neuron. So sensory information below the mandible comes in, touch, I'm talking about, comes in, stays on the same side of where it came in the cord, and goes up towards the brainstem. We're going to pick it up in the brainstem later on, but that's how it's organized. Spinothalamic tract, pain, temperature, crude touch. So if you're operating with spinothalamic tract, if I touch you, Kayla, close your eyes, if I touch you again, say, where did I touch it? You say, well, it was somewhere in here. Being able to crudely localize touch is also carried over spinothalamic tract. But pain and temperature are the predominant sensations. That information comes in and synapses in the dorsal horn. So the first order spinothalamic tract neuron synapses on the second order fiber that crosses in the midline in what's called the ventral commissure of the cord, ventral white commissure, to go to the spinothalamic tract on the other side. So the spinothalamic tract will carry it up. So here's the thing. If Meredith is right here, if I touch her hand, understand that that touch sensation has come in and gone up the same side. If I pained her, that pain sensation came in, crossed, and went up. Note fiber types. Remember we talked about this the other day. C fibers. Oh, you know that pain is coming when you slam your finger in the door. You know it. It's going to take a couple of meters a second to reach it. Ouch. Versus instantaneously knowing, oh, wait a minute, my finger's in the car door. Being the A fibers. And there's our little motor neuron from the lamina 9 going back out to the muscle. There's our nice little reflex arc. It's beautiful. Upper motor neuron. Now, you should have an update here. It was sent out this morning. It's on D2L now. It's on D2L. I did this last night. You don't want to be there with me when I do things at night. That was a snort. That's carried over the dorsal column pathway. When you snort, that's, no, it's not. Let me, I actually have this out of place. So let me reserve, I'll come back to this later on, okay? Let's hold off on that. So, if we look at the sensory fiber types, they're divided into A's, B's, and C's. A's, we've talked, now A's have A delta, A beta, and so we don't, collectively we'll call them A's. These are the large fibers, the very fast fibers. The B fibers have to do with the autonomics. The C fibers are spinothalamic tract, pain, temperature. That's what I've been saying all along, okay? And if you look at these things, here are the A fibers up here. This is a light micrograph. If you look at a scanning electron micrograph, you see these beautiful axons arranged in bundles like this, very, very beautiful. If you look at a transmission electron micrograph, this would be an A-type fiber, and here are the C fibers wrapped up in one wrapping of myelin. Sensory endings for these guys, if you look out in the periphery, the C fibers are free nerve endings. Of course they're in the skin. Of course they're in the skin. They don't have to be in the, they're in the dermis too, and in the subcutaneous tissue, but they're certainly prevalent in the skin. It's the only one really in the skin, would be the C fibers. Because pain and temperature, you want to be able to perceive superficially. Other things like Merkel cells, Pacinium corpuscles, all sorts of bizarre endings are A-type fibers, and they pick up. They have receptors for specific things. Pacinium corpuscles are like an onion. There are layers and layers, so if you put pressure on it, you can activate the nerve ending that's within it. So this would be a type of sensory receptor for touch. Others, it's for vibration. Others, it's for, and so on. Okay? The take-home message here, as far as I'm concerned, is that you should know that free nerve endings are associated with C fibers, pain, temperature. All of the others have some type of specialized ending that is able to detect that particular sensation. These things, they don't generate action potentials. These things do what are called generator potentials. So if I'm, for example, let's just talk about vibration. If something is vibrating a little bit, and I touch Meredith here, and I say, is that vibrating or not? And it is vibrating, but it's vibrating at such a low level, it's not enough. These guys may pick it up as vibration, but not enough for her cortex to determine that it's vibrating. They produce what are called generator potentials. It's like little steps. You get enough generator potentials, and that will cause an action potential to be developed in the process, and then she can say, ah, that's vibration. Generator potentials are a little bit. Action potentials, it's taking the information to the cortex. Right?
There's a delta with a free ending. I didn't know what that is.
A delta.
Oh,
over here?
Yeah.
You know, I don't know what in the world that's all about, because it's a myelinated axon. It's the point I want you to know, though, is that C fibers, pain, temperature are free. Whatever else might be free, I don't care. C fibers are free nerve endings. Now, here's a thing you need to appreciate about the way the nervous system is set up is we talk about it you being like a worm, segmented like a worm. It's not exactly correct. Because if you look at this one primary afferent neuron, or first order neuron, they cut the peripheral process, and what they've done is they injected a tracer into this neuron, and it went into this cord and distributed to all of its parts of its axon. So it went into the cord here at this spinal cord level, but then sent a collateral down to this level and a collateral up to that level. And you see all of these different endings. You see there's the lateral spinothalamic tract right there. There's the, what is that? That's lamina, that's not the spine. That is a reflex into lamina 9, it looks like, right here. Right there. Did it cross the midline? I can't see up there on that thing. It looks like it crossed the midline right here. The point I want to make on this, though, is that one neuron, sensory information coming in one place, distributes up and down the cord another segment or two. So dermatomes are not precise. They're overlapping, and I think I made that point in anatomy, that they're overlapping. Another thing that can cause information to overlap is the fasciculus proprius. Right around the gray matter of the cord, there are neurons that stay within the spinal cord that connect one segment to the next. The fasciculus proprius does that. Where's the name of the fasciculus proprius? Right there. They connect different layers of the cord. So once you get into the dermatomes here, in the central nervous system, you see the overlap. So when you're doing a neuro exam, you're not going to have somebody with sensation at that dermatome and no sensation at the next dermatome if there's a spinal cord lesion. It's more diffuse than that. Right? That's what I want you to appreciate, that it's more diffuse than that because there's overlap of dermatomes. All right, let's take a break and then we'll come back and do the rest.